Mitosis is the division of a cell nucleus in somatic cells, or body cells. It is followed by cytokinesis, which is the division of the cytoplasm. Although the majority of a cell's life is spent metabolizing, not dividing, mitosis is a critical part of the cell cycle to determine its future. Mitosis is what transforms a tiny, single-celled embryo into a vastly complex, multicellular organism like you and I. In each of the various stages of the cell cycle and mitosis, different cellular activities occur. Interphase accounts for about 73% of the cell's life, occurs prior to mitosis, and is also divided into 3 stages. The first stage, G1 or GAP1, contains a regulative checkpoint that commits the cell into beginning the cycle. Next comes S, or synthesis, during which the genetic material duplicates so that each chromosome contains 2 sister chromatids. The number of chromosomes remains the same, however the number of chromatids, which are half of a chromosome at this point, duplicate. The sister chromatids are connected at the centromere with the kinetochore, a bond made of cohesins. This kinetochore forms a hook of sorts for microtubules to attach to and pull apart in later stages. Each time the chromosomes replicate, the telomeres at the end of the DNA strand fray a bit. These ends do not code for genes, but this fraying accounts for aging. The last phase of interphase, G2 or GAP2, contains another checkpoint, after which the cell is committed to dividing. Passing this checkpoint ensures other organelles are duplicated and ready for division, just like the genetic material. Prophase is the first stage of mitosis. Chromosomes condense and are now visible with a light microscope. The centrioles have been duplicated and begin to move to opposite poles of the cell. The spindles approach chromosomes and the nuclear envelope disintegrates. During prometaphase, the microtubules of the spindle have attached to the kinetochores of the chromosomes. Metaphase is characteristic of the chromosomes lining along the metaphase plate. The chromosomes are in a distinct line along the middle of the cell, with chromatids parallel, ready to be pulled by the microtubules to opposite ends of the cell. However, cohesins have not yet released at the centromeres. Finally, during anaphase, the sister chromatids are pulled apart to opposite sides and centrioles. Lastly, the cell divides into 2 during telophase and cytokinesis. The nucleolus and nuclear envelope reform. In animal cells, the cleavage furrow forms, deepens, and eventually creates 2 distinct cells. In plant cells, a dividing cell wall is built with cell plate fragments. Thus, 2 daughter cells have been formed, nearly identical to the parent cell, with the same number of chromosomes. In this case, the diploid parent cell forms 2 daughter diploid cells after mitosis.
Meiosis is very similar to mitosis, but a diploid parent cell undergoing meiosis results in 4 haploid cells. Meiosis is critical in sexually reproducing organisms. For sexual reproduction, a maternal and paternal haploid cell fuse to form 1 diploid cell, which will then undergo mitosis to grow. Without meiosis, each generation would have 2 times as much genetic material as the previous generation, which would obviously cause problems. Meiosis is divided into 2 primary stages, 1 and 2, and each stage is further subdivided into the same 5 phases as mitosis: prophase, prometaphase, metaphase, anaphase, and telophase cytokinesis. Meiosis 1. Meiosis 1 occurs like mitosis with several exceptions. Prophase 1 is very similar. Chromosomes condense and crossing over occurs. Crossing over takes place only during meiosis, not mitosis at the chiasmata. Small bits of DNA on the homologous chromosomes are exchanged by non-sister chromatids. This is called recombination and accounts for more genetic variety. In metaphase 1, the homologous chromosomes, not sister chromatids, are lined up parallel to each other on the metaphase plate. The result of crossing over holds the homologous chromosomes together and stationary on the metaphase plate. Therefore, in anaphase 1, homologous chromosomes are pulled to opposite ends. Both telophase and cytokinesis occur, just like in mitosis. Meiosis 2. However, another difference occurs in restarting the cell cycle for meiosis 2, because the chromosomes are not duplicated. The cell cycles through prophase and prometaphase again, and reaching metaphase 2, the chromosomes line up just like in mitosis. In anaphase 2, the sister chromatids are pulled apart, telophase and cytokinesis occur again, resulting in 4 haploid daughter cells.